like to welcome you all to this webinar on developing a therapeutic response to COVID-19. We have understandably had a tremendous amount of focus on accelerating the development of vaccines, but here we're going to focus on the therapeutics that could potentially come to market and help improve outcomes for patients with COVID-19 before vaccines will be available. There are a number of treatments in development now. Today, we're going to try to focus on a set of issues related to these treatments, including prioritizing and advancing therapies that look promising from a preclinical standpoint, how they can get through preclinical testing as quickly as possible. Then in the clinical testing phase, making sure that we are bringing together the most efficient and rapid ways of undertaking clinical testing for what may be a significantly greater number of compounds in testing in the months ahead than we're seeing right now. Also important is building capacity for manufacturing and production. At every step of the COVID-19 pandemic, we have struggled with issues of the scale of the pandemic and bringing our response to scale, and that may be the case for developing clinical treatments as well. And then finally, since many of these treatments will either get to the market under accelerated pathways with significant but limited evidence on effectiveness in a variety of treatment conditions and for a variety of patients, and because many of the treatments that may become available include treatments that are on the market now, the development of better evidence on COVID-19 treatments in the actual real world context is also critical for responding to the pandemic. So we're going to try to cover all of these topics today. We're going to kick off with a discussion with my former colleague, former FDA commissioner and current resident fellow at the American Enterprise Institute, Scott Gottlieb. From there, of a fireside chat with Francis Collins, the director of the National Institutes on Health, who just had a very important announcement on these topics on Friday. And then we're going to have three workshop sessions. The first on accelerating COVID-19 therapeutics, drawing from the experiences of some of the leading innovators in this space today. The second on strategies for developing, accelerating, and conducting clinical trials on promising therapeutics and getting those to patients as rapidly as possible. And then the third on the potential role and expanded role of real-world evidence to improve our understanding and use of these therapeutics in practice. We'll end with some closing remarks. Just a couple of brief logistics before we get started. This webcast is being recorded, will be available on the Duke-Margolis website following the event. All the participants are going to be muted during the webcast. If you have a question or comment, please use our hashtag COVID Therapeutics, COVID Therapeutics, all one word, hashtag COVID Therapeutics, and we will work your question or comment into the discussion as best we can. So I'd like to get going right now by introducing Scott Gottlieb. Scott, thanks for joining us here today. You've spoken a lot about the urgency of making progress on therapeutics, especially these hopefully shorter term alternatives to vaccine development. We've worked on some joint reports on this topic together. Where would you like to start? What are the most important things for people to think about as we're trying to work on bringing these therapeutics to patients as quickly as possible?
I'll take about 5 minutes just to offer some top-line thoughts, if that's okay. I think it's important that we have something available for the fall. I think hopefully as we restart getting people to work in May and June, gradually get a transition back to some semblance of normal life, we can quell the risk of major outbreaks or an epidemic as we head into the summer. I think the hope is that there is going to be some seasonal component to this virus, and we'll do this gradually enough and hopefully get a break from the hot, humid months in July and August, which might be a backstop against epidemic transmission. Droplet transmission is less efficient in the hot weather. The epidemiology of spread changes, so people aren't close together indoors anymore in the summer. Hopefully all of that gives us enough of a backstop, coupled with our own prudence, that we can come off this epidemic safely in the summer. I think that there's a real risk that as we come back in the fall, schools back in session, residential college campuses are back in session, people are back at work more earnest and trying to travel again for business in the fall, that we face a risk of outbreaks or maybe another epidemic again. And then the question becomes, what's our toolbox? Why would this time be different? And that toolbox is going to be much broader screening, but hopefully it's going to be one or more therapeutics that could help be used both as a treatment in early disease and an effective treatment to help mitigate some of the risk of the virus, but also as a prophylaxis. And when you look at the universe of treatments, there's really only a small subset of treatments that could potentially be available. It's the antivirals currently in development, most of which are just being repurposed against COVID-19, as well as the therapeutic antibodies that are being developed by 4 companies, Amgen, Lilly, Vir Biotechnology, Regeneron, that could potentially be used as, could be potentially available by the fall. The antibodies could be used as both a treatment in early disease, as we saw with Ebola, as well as an effective prophylaxis and a bridge to a vaccine. The antivirals probably will have most of their benefit if they're used earlier in the course of the disease to prevent disease progression. And remdesivir is obviously the one that shows potentially the most promise and is the furthest ahead in clinical development. So it's a pretty small toolbox. And I think it's incumbent upon regulators around the world to focus on the products that could be available and try to pull them through development. We need to be focused on everything that could potentially be available for COVID-19, but there's really a small subset that could be available in time to mitigate the risk in the fall. And I think we have to focus particular attention on those. So what does that look like? I would break it down across 3 domains: the clinical trials that we're looking to run, issues around manufacturing, there's issues around access. When it comes to clinical trials, I think we need to be mindful of a number of things if you're thinking about the fall. As we start to put these drugs into broader clinical development in the summer, the epidemic's going to be subsiding, hopefully. And so we're going to be testing, for example, the therapeutic antibodies probably in July and August at a time when infection rate here is declining. So I think it's incumbent upon us to think about how we do that. You saw with the Ebola clinical trials done in West Africa that it was very hard to enroll those trials in a setting where the epidemic was declining. The same thing became apparent in China recently with COVID-19. So thinking about how we develop protocols where these can be tested in other regions of the world that might be going into epidemic spread, and do that in a way where, we're going to have to address the issues of, will we supply the drug in those regions if we're going to be conducting clinical trials in those regions? Those are going to be issues we should start grappling with right now. Because, for example, testing therapeutic antibodies and looking at their ability to serve as a prophylaxis in a setting where infection rates are declining, hopefully sharply in July and August, and background spread is much lower, that could be difficult, especially when the groups that you'd want to test it in who are at higher risk of contracting the infection might already have high background rates of seroconversion, so they might be less prone to develop the virus and not be good candidates to put a prophylaxis in. In fact, they'd be probably contraindicated from receiving a therapeutic antibody if they have natural antibodies. Same thing holds for a vaccine. We may have a lot of doses of a vaccine available by the fall. The question is, how would you test it in the setting of an epidemic? I think that there would be potentially tens and maybe hundreds of thousands of doses available at some point in the fall. If you have a major outbreak in a city, you might want to think of things like a step wedge, a cluster randomization, where you would randomize people in a city to receive a vaccine, but in different cohorts. So you might give one group the vaccine initially, 25,000 patients initially, and then 2 weeks later, another 25,000 patients, 2 weeks later, another 25,000 patients, and basically use the different cohorts to randomize against each other to look at and see how the vaccine is working. So you would provide the vaccine under a protocol, but also be delivering it in a setting where hopefully it could help mitigate the risk of epidemic spread. That's something that we need to be thinking about right now, whether we would reach for those kinds of trial designs if in fact we have large outbreaks. The second domain on manufacturing, and you and I have talked a lot about this, Mark, and you wrote a lot about this in the paper. I think there's a lot we can do to try to scale up manufacturing capacity in advance. It's a little bit more difficult with the vaccines, although the companies are doing this at risk because the facilities aren't interchangeable. But certainly when it comes to the therapeutic antibodies, we could think about how we develop some common manufacturing capacity that could be loaned out to whoever's first to market with a therapeutic antibodies that were not supply constrained at the time that these become available, hopefully in the fall. And so I think that there's an opportunity to do that right now, to move products that are being manufactured in the U.S. out of U.S. facilities into European facilities to free up domestic manufacturing capacity. All of this kind of industrial planning needs to be going on, and it's probably beyond the capacity of just one company to think these issues through. I think there's a real role for the government to try to coordinate this and try to invest resources. And Congress explicitly made those resources available in the CARES Act, where they set aside over $10 billion for the development of advanced manufacturing and scale up of manufacturing so you can have commercial scale supply on hand at the time of approval. And I think it's incumbent upon us to think of an organized way to try to quickly stand up these kinds of opportunities. And then finally, on access, I think that if you look at what Gilead's doing with respect to how they're providing remdesivir, I think it's frankly a model, even though it's come in for some criticism. There's a rigorous trial underway with NIAID that's going to allow us to turn over a very important data card on whether or not that drug's working and what the true scope of the treatment effect is. But in addition to that trials, the companies stood up a number of open label studies. And one is in moderate patients, 400 patients in a moderate trial. Another is in the severe setting, where they've consistently expanded the scope of that trial. And they're using the trial to both collect information as well as provide access to the drug in some kind of protocol. And that's what we've long wanted to do, instead of just providing drug in a expanded access fashion where you're doing, individual INDs for patients and not really collecting information that can be useful in informing clinical practice. Gilead's actually collecting information in the context of those open label studies. They're randomizing to 2 different doses, and that's going to give them some incremental and important information in addition to filling out the safety database. And so I think we need to recognize what's being done in that context. There was some criticism of, well, these are open label studies, but I think that was by people who didn't really recognize that alongside those open label studies, there was a very rigorous study that's been expanded by NIAID. That's a randomized study that's going to rigorously answer the question about the scope of the treatment effect. And so I think in some ways it's sort of an optimal approach in the setting of an epidemic like this. You have the rigorous trial underway to definitively answer the data question while you're collecting incremental information in open label studies that are being run alongside that, and not impinging your ability to bring that randomized trial to completion. That randomized trial is fully enrolled now, as best I understand. So I think it's worth looking at that as a model for how we do trials in this setting. I'll pause there, Mark, and appreciate the opportunity to join today. Thanks a lot.
That's a great overview, Scott, of the topics that we want to cover today. Just a very quick follow-up, and then I want to turn to Francis, who's taking some important steps on many of the topics that we just discussed. And that is timing. So we're talking about treatments on an accelerated pathway with all of these efforts underway that may be available by fall. We'll come back during the rest of the session to that time frame and ways to perhaps accelerate it. But you're really saying that we need to focus on issues like manufacturing capacity and thinking about how additional evidence might be used in the real world setting from emergency access and other mechanisms right now.
So yeah, I'm glad to weigh in here, Mark, if you want to begin this fireside chat minus a fire to see everybody. And Scott, that was a tour de force, as you're famous for, packing an awful lot of information into just a few minutes of a summary there. I do want to say a bit about this new partnership that was announced on Friday that's been in the works now for all of like 2.5 weeks, called ACTIVE, which stands for Accelerating COVID-19 Therapeutic Interventions and Vaccines. This partnership has engaged 16 pharmaceutical companies, the NIH, the FDA, the CDC, BARDA, the European agency that's the equivalent of FDA, and is being managed in terms of its project management by the Foundation for NIH, building on the success they've had in the past with the Accelerating Medicines Project called AMP. So this tries to address a lot of the issues that Scott just raised for both therapeutics and vaccines. I think we're talking specifically today about therapeutics, so I'll focus on that. We are delighted that the remdesivir trial has been going forward. You mentioned that. It actually over-enrolled. It just stopped enrollment a couple days ago at 900. It was powered at 500. And we could have an early look at that even in a couple of weeks to see by the DSMB view whether there is some signal there. And certainly in a month, probably, we should have a pretty good idea of whether there has been benefit. And so that's coming pretty soon, and everybody will want to watch for that. My own bet is that if it shows benefit, it won't be everything you need. And so we will still wish for other kinds of therapeutics to add to that. If HIV takes more than one drug, I'm not sure why we think coronavirus, SARS-CoV-2, will be cured with one. We may very well have a circumstance where we need to think about combination therapies. So what are we doing about that? If you look at the inventory of potential therapeutics that have been suggested by somebody, and that are actually things that have already been in humans, so they could be put immediately into a phase 2 trial. Now, depending on which inventory you look at, there's more than 100 of those, and there might be as many as 600. And they're in various places. BioCentury has such a database. Milken Institute has a tracker that has a lot of those as well. NIAID has been also accumulating similar things. So first thing we need to do, which is what Active's working group on clinical therapeutics is now focused on, is to sift through that inventory and try to decide what are the highest priority therapeutics that really should be at the top of the list for clinical trials. That is underway right now. And of course, what you would like to have are compounds which have a decent scientific background, where you know the mechanism of action, and where manufacturing capacity is not going to be a problem if you happen to get a good result in a small trial. Those kinds of criteria are being explored by this group. Again, about half industry people, half academics and NIH people. And I'm pushing them pretty hard to try to come up with at least a first pass at what you would say would be the sort of top 6 to 8 candidate therapeutics that ought to go into trials next. And I would want those not all to use the same mechanism of action, because what you'd really like to do is to have different mechanisms of action, and you pick sort of best in class for each of those and then try to get them into the queue. I will say pharmaceutical companies have been completely endorsing this effort and not arguing that it's got to be their compound. I've even heard a leader of a particular large pharmaceutical company say, you know, if it's my competitor's compound, but I've got the clinical trial capacity to test it, I'll do it. Everybody just wants to get this done. And that's really gratifying to see the way in which this crisis has brought people together in exceptional ways. The other thing we need to have, of course, if you're going to do this, is a really organized clinical trial capacity system, so that if you had those top 6 or 8 compounds, you would immediately know how you could slot them into trial networks, and you would know how many patients of which degree of severity those networks might have access to. And that is another working group that is actively trying to pull all of that together. Some of those are NIH clinical trial networks. Some of them are things like PCORNet. Some of them are industry run. They've never really been synthesized in this way or tried to organize in such a way that you could predict over the next 3 months how many patients with COVID-19 of what degree of severity might be available for trials. But we got to have that if we're really going to do this in an orchestrated way. So that's underway. Both of these are together working on the need for a master protocol so that we don't end up spinning our wheels by putting things together very slowly. There's certainly a possibility here of having a common control arm for different kinds of therapeutics, and other kinds of designs using adaptive trials are very much in the works. FDA is deeply engaged in Active in all of these working groups, and that's really important, so that whatever we come up with will in fact meet regulatory standards that they'll approve of. And obviously, we're partly talking about antivirals, but we're also talking about immunological approaches, antibody approaches, and we're also talking about the therapies that might work best for people with advanced lung disease, where we think potentially the overreaction of the immune system, maybe it's a cytokine storm or something like that, might be a totally different kind of target for therapeutics, and probably might be something you wouldn't want to give to somebody with minor symptoms, but might be just a thing for somebody on a ventilator. So there may need to be obviously different considerations, different master protocols, depending on the severity. All this is moving at rocket paces. The first organizational meeting of Active was on April the 3rd. Here we are now. The 4 working groups have all met multiple times already. There will be a feedback to the parent group coming up on Wednesday to say where progress is, including timelines. Most of the people involved in this, which is about 50 from different backgrounds and different expertise, have pretty much dropped everything else to try to make this happen. BARDA is a critical part of this because, as was mentioned by Scott, they have $10 billion that can be applied for therapeutics and vaccines in order to do the manufacturing scale up if we know what we think is a good bet. And again, the idea is not to wait until you're sure to make that bet, but actually to start scaling up for things that even look like they might work, so that you don't end up with a delay at the end. So all of that sounds good. Will we be at the point by July or August where we've actually demonstrated 1 or 2 or 3 therapeutics that have been documented to be successful in a randomized controlled trial? I don't know, but we're sure going to do everything possible to try to make that dream come true, because that is so critical for what we're going to be facing in the fall, as Scott very much put forward. I even wonder, this is just a little final comment, that maybe this could also be helpful in terms of vaccines, because if you had a really effective therapy that you know was pretty much going to work in a healthy young person, we might be able to begin to shift our vaccine demonstration projects into volunteers instead of expecting that we're going to do this in a very large-scale phase 3 trial. That would be a wonderful way to short-circuit the regulatory definition of what means safe and effective. So with that, Mark, back to you.
Francis, thanks for that great overview. And it does sound like there's a tremendous amount going on already, certainly unprecedented. So great to hear about that. Let me ask one question. I think Scott may have a follow-up for you too. And that is about how this prioritization process is working. Totally appreciate the need for that. And NIH generally does that when it prioritizes grants. You mentioned BARDA. It does that when it prioritizes emergency countermeasures, and FDA has fast track, breakthrough designation, and so forth. But it seems like what you're talking about here is going beyond that, like actually helping with preclinical testing in a more extensive way. We've talked already about the potential concerns about adequate access to well-designed clinical trial networks in a period when the virus may be active at a much lower level than we've seen in recent months. Can you say a little bit more about what this special assistance is likely to involve as you see it?
Well, again, I think what's totally different about it is bringing all the stakeholders together around the same table to try to do this prioritization. So about half the people who are doing this are not government types. They are from industry. They have to leave their favorite hobby horse at the door in order not to have this be a conflicted situation. But they basically have agreed upon what the criteria are for that prioritization and are now rolling up their sleeves to start to go through the data to say, of the candidates that we've got out there, which of those would rise to the top. I'm sure that will be an area of some disagreement. It should be, because a lot of the science is imperfect, but I don't think we've ever tried to do anything quite like this before. I am sure that in the dust it all settles, we'll figure out we made some mistakes. Maybe we passed over some sort of long shots, some diamonds in the rough that didn't look very promising and ultimately someday turned out to be. But it seems like this is the best we can do right now with the subject matter experts from every sector getting around the same table to sort of hash it out and then holding ourselves accountable for testing those particular candidate therapeutics.
Scott, you got an additional perspective or thought to add based on these further steps that we've just heard about?
Oh, look, I think that this is the exact thing that that the government can and should be doing. So I applaud Francis and NIH for the leadership that they're showing here. I think we're dealing with, so I'll just sort of close here. We're dealing with scarce resources when it comes to the ability to conduct these studies.
Yep.
And I think that this is a time to try to bring together collective action to define sort of an efficient platform. And frankly, it's going to require us, I think, government officials, and I'm not giving instructions to Francis or anyone, it's just my opinion independently. I think it's going to require government officials to do something that typically you don't do, which is try to pick some winners and losers at the outset, or certainly some winners. Not necessarily pick losers, but try to identify what you think is most promising that can be available in a reasonable time frame, and focus on those. Because if we focus on everything, we're not going to have anything potentially, and we have very scarce resources to try to pull through not just the clinical trials, but also the manufacturing as well. And so I think we should be trying to focus those resources on opportunities that can deliver the most potential benefit in the shortest period of time.
Thanks. That's exactly what I'm charging this working group to do. It's not an easy task, that's for sure, but I think they're the right people to take the best shot at it we've got. And you're right, manufacturing has to be a part of it as well. It won't do us any good to find something that looks really promising, but you can't make enough of it to help people.
Yeah, and those manufacturing constraints could be important given the scale of the epidemic and the need for broad global treatment. Francis, just to follow up, Scott gave the view that sees only a limited number of compounds potentially getting to advanced testing by summer, and therefore potentially on path for broader use in the fall if all of these steps we've just been talking about go right. Do you see, from what you've come across already in this process, any promising additional mechanisms, types of compounds beyond those that we've already talked about? So some of the lead antivirals, and I think we agree with you too that if you look at other conditions, HIV, et cetera, you start out with treatments that are not going to cure everybody quickly, but that make some progress. You build on that knowledge to expand the options and maybe get to combination therapies. Here we've already talked about monoclonal antibody therapies, and our next session is going to focus a lot on that. And you mentioned treatments for the immune response that may get overblown to a very damaging degree in some patients. Is that the spectrum? Are there other things that we should be thinking about or that you're starting to explore?
There's so many things we'd love to think about if we had time. There is another part of the active effort on preclinical approaches, but it's mostly just looking at things that have already been in humans for totally different reasons that might potentially be repurposed, which means you need some validated assays so you could check that out on maybe tens of thousands of compounds and see if you might have a winner in there somewhere. And who knows, if you were successful at that, you might find something that had a totally different mechanism of action you didn't expect. I think, though, for the timetable we're talking about, mostly what we are going to have to look at are the kinds of things that you've already mentioned. I will say there are going to be some surprises. The recent, very small, very open label trial using acalabrutinib, which is a expected BTK kinase inhibitor, showing what appeared to be pretty good results in people with advanced lung disease. And now that's started an AZ trial of a randomized sort. I guess I would not have guessed that some second generation ibrutinib compound would ever find a place in COVID-19. But I guess that shows I don't know a lot about biology after all.
Well, it's complicated biology, and hopefully we'll get some more good surprises like that in the weeks ahead as a result of efforts like Active. Francis, thanks for joining us. I expect you've got a lot of those work group meetings and other activities to get back to. We really appreciate your taking the time to be with us. And I'd like to thank you.
Thank you very much, Joe.
Great. Transition now to our first panel on accelerating therapeutics, and very pleased to have joining us for that George Scangos, who's the CEO and director at VIR Biotechnology, George Yankopoulos, who's the founding scientist, president, and chief scientific officer at Regeneron Pharmaceuticals, and Chad Robbins, the chief executive officer and co-founder of Adaptive Biotechnology. So George, and Chad, we'd like to maybe start off by you all telling us a little bit about what you see, kind of adding to the discussion that we just heard about the best opportunities from your company's perspectives, the products that you're developing, and any special challenges or notable issues about the path forward from here. And I'd like to focus on, since we've talked some about antivirals and we're going to get to maybe some of the immune treatments, I'd like to focus on the monoclonal antibodies. That is a area that has gotten a lot of attention, viewed as very promising, kind of a new, it's been used in Ebola, but kind of a new approach for a global viral vaccine pandemic like this one. And maybe George Yankopoulos, could I start with you and some of the activities underway at Regeneron in this area? And we may not have George on with us right now. That's all right. While we sort that out, let me go to George Scangos with the same question. George, good to see you.
Okay, thank you, Mark. I can tell you a little bit about our approach and what we've been doing and what we see as the challenges going forward and our thoughts to how to deal with those. So VIR's about a 3-year-old company, completely focused on infectious diseases. The whole purpose for starting VIR was that we saw a huge need for a really well-funded and aggressive biotech to tackle some of the major public health issues around the world that are caused by infectious diseases. We jump-started ourselves. We made a number of acquisitions, signed a number of collaborations. So among the companies that are now part of VIR was a company called Humabs, which was in operation for over a decade. isolating antibodies from patients who'd recovered from diseases. So Zika, dengue, malaria, go on to the list of pathogens. We believe that the best antibodies have breadth of coverage, and that the breadth of coverage correlates with the difficulty that a virus will have in escaping the antibody, because any epitope that's been conserved over many years of divergent evolution, when most parts of the virus have changed, must have constraints on it and is likely to be very important for the virus in some functional sense, and therefore very difficult to mutate. We did that for flu. We have a pan-flu antibody, flu A, that neutralizes every strain of flu that's arisen since 1918. Flu A, let me say. So all the pandemic strains, all the seasonal strains. That antibody was about to start phase 2 trial in the Southern Hemisphere flu season this spring to see if we can provide a higher level of protection for high-risk populations than they get through the vaccine. Of course, that trial is postponed pending COVID. Specifically for COVID, we went back after the SARS epidemic. We had isolated a number of anti-SARS antibodies and had banked a large number of B cells from patients who'd recovered from SARS. And then we went back and I think 9 years later, isolated additional B cells from SARS patients, wanting to understand whether the affinity maturation that could occur over all these years would yield better, more potent, more diverse antibodies. We've now screened those antibodies against COVID-2. We have identified 25 antibodies from SARS patients that cross-react with COVID-2. We prioritized 7 of those, and one of them, which we called S309, is now being manufactured. It is a potent neutralizing antibody against both SARS and COVID-2 and hits an epitope that is highly conserved across this whole family of not all coronaviruses, but the subfamily of coronaviruses to which SARS and COVID-2 belong. It is, that epitope is, let's say if you look among all the divergent members of that family, there's 24 amino acids in that epitope. 19 of them are absolutely invariant. The rest are all very conservative substitutions. Among the 8,700 or so isolates of COVID-2 that have been sequenced, that epitope is invariant. We found one conservative change that we're testing, but we don't think that will have any indication. That antibody is being manufactured in 2 versions. Both versions have a half-life extended mutation in them. One version has an additional alteration in the FC region that enhances its binding to FC gamma stimulatory receptors, 2A, 3A, and essentially eliminates binding to inhibitory FC gamma receptors. And so in model systems, that alteration increases the near-term potency several-fold and results in the induction of T-cell immunity that provides an additional level of protection. So those antibodies are being manufactured. We'll start clinical trials, as alluded to, this summer. Just talking about some of the issues that you've alluded to, we don't know where the epidemic will be in what stages in the summer and fall as we do the testing. And so we are preparing certainly to do the trials in the US. It may be that other regions of the world have more appropriate numbers of patients. And so we are also gearing up to do the trials in other geographies as well. We had a number of sites geared up in Australia, New Zealand, South Africa to start our flu trial. It would be easy enough to go back to them and do the trial in the Southern Hemisphere if the epidemic is more prevalent then. We've thought a lot about the manufacturing, as you've alluded to. It doesn't, probably the only thing that's worse than not having a therapy that works is having one that works and not being able to supply it. And so we've lined up a lot of manufacturing capacity. We think even by the end of this year, we can have hundreds of thousands of doses, potentially more than that. And going into next year, we'll be able to have tens of millions of doses ready through the manufacturing agreements that we've put in place. So unfortunately, you have to do all that at risk because we're in such a short timeline. And so you can't wait until you know how well your therapy works. So we've lined up a lot of manufacturing capacity at risk, with the assumption that if for some reason our antibodies don't work, somebody else's antibodies do, that manufacturing capacity will be used by somebody, right? And whoever has the best therapy will be able to utilize it. So that's where we are. We have additional antibodies that, let's say, also bind the receptor binding domain, but at different epitopes that can be used in combination with our lead antibody. And so we're bringing those forward as well. If a single antibody can provide an effective therapy from which the virus can't escape, it has a lot of advantages in terms of cost of goods, in terms of the number of patients who've been treated with a given manufacturing capacity. Then that may or may not be the case. And so if we have other antibodies we're bringing forward that we can use in combination if we need to. We've designed adaptive trials so that those additional antibodies can just be slotted in without time loss into the existing trial. And we'll test both two versions of our lead antibody as well as a lead antibody in combination with others to see which provides the most effective therapy and the most effective prophylactic regimen.
Great. George, thanks for that overview of the path from where you are now, thinking ahead all the way through clinical testing and manufacturing. Chad, maybe we could turn to you next at Adaptive. You're also working in the antibody space on actually on diagnostic as well as therapeutic, though we're focusing on the therapeutic side today. How does this pathway look from your standpoint? Any other key issues or challenges you'd like to highlight?
Yeah, sure. I actually will. I'll touch a little bit about what we're doing on the diagnostic front, because I do think it has some relevance. But first, I want to thank Joel and Lynn from Alexandria and Mark and your entire team at the Duke-Margolis Center for Health Policy for bringing really this awesome group together so that we can learn from our respective and collective experiences and expertises to combat, which is, truly an unprecedented health crisis. And also, I have to say, I'm honored to share the virtual stage with George Skangos and hopefully George Duke-Margolis will be joining as well, which two legendary drug developers in our industry who are tackling COVID-19 from their unique perspectives. And as Francis mentioned, I want to echo the sentiment. It's been incredibly motivating to see everyone come together to solve these global issues, both in the industry side and the government side and the policy side. With respect to what adaptive technology can contribute, we can decode the immune response data to COVID-19 at a massive and unprecedented scale. This data can advance our efforts to both better diagnose and treat the virus. And so we're applying this data in two important ways on both the diagnostic and on the therapeutic fronts. Starting with the diagnostic side, we were expanding our existing partnership with Microsoft to transform this massive and unprecedented immune response data to COVID-19 to create a diagnostic that will provide a full picture of a patient and a population's immune response. More specifically, what we're doing is we're measuring the presence of specialized cells in the immune system called T cells. Actually, we're not measuring antibodies. We're measuring the T cells that can identify the disease early on and then be able to clone themselves to be able to combat the infection. I think there's been great progress that's been made in a short amount of time in PCR-based tests that look at the virus itself, and also the serology tests that are looking at antibodies, but they're only looking at a partial antibody response. And there's still issues both on standardization of these tests, and there's biological issues. As we continue to read about every day, another great article in the New York Times yesterday, it's really making it hard with these tests that are on the market and coming on the market alone for us to understand, one is how widespread the virus really is, and two, who has truly developed immunity to the virus. So what our team is doing is cranking on a cellular immune test that may resolve some of these issues, a test based on the T cell receptor response to the virus. And we think, I think we all know this is going to be critical to solve some of these issues so that government entities can set these policies to allow us to get back to work and the economy to reopen. We hope to have data on this shortly, and importantly, and I think this is crucial to point out, is we're going to be making this data publicly available to public health officials, academia, and industry, not just to advance adaptive tests, but in the collective good, so that others can use our data to make their own contributions to COVID-19.
I really appreciate that emphasis on data sharing. That goes along with the themes that we've been discussing already. And again, I think it's kind of unprecedented. Now, I think we may have George Yankopoulos with us. If we try to unmute the phone line, hopefully get this to work. I know George was trying very hard to be on with us. See, are we able to unmute?
Can you guys hear me?
Yes. There you are. Great. We can even see you, George. Looking good. Thanks for being with us. We really appreciate it. Hopefully you're able to hear some of the preceding discussion. Just a similar question for you. Where are things at Regeneron? I know there's a lot going on there, and what do you see as the challenges and opportunities going forward that you'd like this whole group to hear about?
Right. Well, I guess you've heard a lot about antibodies already. You heard from the other George that they went back and they looked at SARS-1 patients and tried to get cross-reacting antibodies. Well, as you guys know, we have probably the most powerful and most rapid response technologies in the industry. So we actually went directly for SARS-2. So we have two unique technologies that probably don't exist anywhere else. We have these humanized mice that make fully human antibodies that we can challenge with the SARS-2 virus or with components of it. And we can also use a very related technology to go after SARS-2 recovered patients as well. And so we employed both of those approaches. We went after specifically highly potent antibodies against the SARS-2 and against the SARS-2 receptor binding domain, the most conserved, important domain of the so-called spike protein that's required to bind the famous ACE2 receptor for the SARS-2 virus. And we were able to very rapidly come up with thousands and thousands of antibodies, both fully human antibodies from our genetically humanized mouse platform, as well as from human survivors themselves. These are antibodies as powerful and as potent as have been described against any antiviral approach. Unlike what George described, his cross-reacting antibody, it doesn't actually bind to the receptor binding domain, the key part of the virus that's most conserved that binds to the human host receptor, the ACE2 receptor. All of our antibodies were designed to target that most critical domain. And as I said, we got thousands of them, and they're also, just because they're so specific in the power of it, at least through cross-study comparisons, they're orders of magnitude more potent than these cross-reacting type of antibodies that George described that you can get from SARS-1 patients. And we were able to, because our technology allows us to do this, we put many of these in manufacturing-ready cell lines. And we've actually already selected a collection of cocktails. We believe cocktails are critical, that you don't want just one antibody, you want multiple antibodies just in case the virus mutates. So we put together several different cocktails, and we've put them all in our queue for large-scale manufacturing. We've also already tested them, since we have such high throughput approaches, we've tested them against all the known variants of the SARS-2 that has been identified and publicly released in the public domain. And we've chosen, since we have such a large collection of antibodies, we were able to select for our cocktails antibodies that we're still able to bind very potently and neutralize and inhibit the virus for all of the mutant strains that have been seen to date. So this gives us an incredible now collection of antibodies in these cocktails. And because of the speed by which we're able to produce these are on schedule to go into human trials in the beginning of June. So basically we can, we're hoping, and we have a variety of collaborations and collaborators that we're talking about, of testing at least the first of our cocktails in 3 sets of trials. We're going to be testing it in prophylaxis trials, that is people who have yet to be infected to show we can protect them, in patients who are suffering from early disease, that is diagnosed, the sort of patients that come to the ERs or seek medical care but are sent home because their disease is not advanced or serious enough. Unfortunately, we know about 20-30% of those patients come back with serious disease. So that's our early treatment population. And we're also going to be going after a late treatment population. As you may know, these are the people who are most seriously affected, who have trouble breathing, who are in the hospital. Many of them are on ventilators. As you may know, we, in collaboration with the FDA and with BARDA and with New York State and with others in the federal government, initiated very rapidly a study in these late treatment patients with our IL-6 receptor antibody, which is another near-term hope that we and others have that might be able to impact this crisis. So basically, where we stand is, as I tried to summarize, we have our very potent, very specific to SARS-2 antibodies against the critical domain of the receptor that are already being scaled up, and hopefully, if all continues to go on schedule, will be in human trials by June. And we also have a completely separate effort where our IL-6 receptor antibody, which was parenthetically derived using the same exact technologies, but old versions, it's about a 15-year-old antibody, is being tried based on some uncontrolled studies that were done in China that claim that blocking the IL-6 pathway might have benefit for at least some patients with this disease. So those are the 2 sets of efforts we have. The antiviral antibodies, which are really moving along very rapidly, as well as trying to test the China hypothesis that the IL-6 pathway is really important.
Yeah, great overview, George. I want to go back to Chad to especially focus on this maybe multimodal or multiple types of antibodies that are being developed and seem to hold considerable promise. Maybe you could, Chad, anything you'd like to add on that, and then I'd like to get to one more question about the about the monoclonal antibodies. And that is, aside from their use in patients who are infected, we talked earlier about the importance of prophylaxis, and that might be at a stage where they're lower exposure to the virus. Are they going to be as effective, or do we think they'll be as effective in that context? But Chad, maybe you first, then we'll stick with this a little bit longer, and I want to try to get to 1 or 2 other topics quickly.
Sure. Sure. Thanks, Mark. And like George Duke-Margolis, we're also going directly after SARS-CoV-2. Therapeutically, what we're doing is we're analyzing kind of this massive amount of immune response data to really look for the Michael Jordan of antibodies, which could neutralize the virus. We don't have development and engineering expertise, so we're partnered with Amgen to identify and develop therapeutic antibodies from the blood of patients who are either actively fighting or who have recently recovered from COVID-19. As George Scangos mentioned, we think that neutralizing antibodies as well, in general, can be an effective therapy, since this virus seems to mutate more slowly than other RNA viruses, and we believe that the mutated strains are genetically similar. So potentially one or a combination of antibodies is a really good therapy for this particular virus. We've also seen some kind of early reports of success using convalescent plasma therapy to boost the ability of patients with severe cases of COVID-19 to fight the infection. And obviously this is great, but unfortunately it can't be scaled, it can't be standardized, nor can it be quality controlled. With Amgen, what we're able to do is use our high throughput way of screening immune cells to find the best antibody or antibodies. These could be used to treat patients fighting the disease. And to your point about prophylaxis, these could be, this antibody approach as a temporary immune boost could also potentially prevent disease in those with heightened exposure, such as healthcare workers. It's clearly it's early days for us, but we're already moving fast. We're seeing some really exciting results. I'll talk more in the Q&A about it, but we're pretty confident that we can deliver really potent antibodies to Amgen in the coming months for neutralization assays.
Great. I'd like to go to one further topic that came up earlier, and that's emergency access. Obviously, getting well-designed clinical trials done as quickly as possible is key to learning which treatments work best and providing a foundation for making available effective therapies. But this is a condition with no current effective therapies. And so tremendous unmet need, lots of pressures we're seeing for access to treatments before we've demonstrated safety and effectiveness. Maybe George Skengos, I'll start with you, but if you or others have any comments about how to do expanded access or compassionate use right, getting the trials done, but also maybe learning from the emergency use or expanded access context Scott Gottlieb described earlier. Do you have a strategy on that? Is there any thoughts about the best way to handle both access and learning quickly?
Sure. Thanks, Mark. Let me, I will address your question. I did want to make a couple of comments to make sure people understand accurately where our antibodies come from. So first of all, the SARS antibody is highly potent. It binds with picomolar affinity, so hard to get much more potent than that. Secondly, we have a whole other series of antibodies from SARS-2, CoV-2 patients that bind to different sites. They all bind to receptor binding domain, different epitopes within the receptor binding domain. So, and those are highly potent as well. So I think my hope here is not that we get into whose antibodies are better, but that I hope everybody's antibodies work, because we might need a capacity around the world that no single company can demonstrate. We are struggling with that concept as you are, right? The goal is to get these antibodies to as many people as we can, as quickly as we can, and to do so in a scientifically rigorous and medically responsible way. And we are thinking about 3 trials, just like George Y alluded to: prophylaxis, patients early in the disease, patients who are hospitalized. So essentially same patient populations. And you could think about emergency use differently, I think, in those 3 populations. Obviously for prophylaxis, and we're thinking both pre-exposure prophylaxis and post-exposure prophylaxis. So you can take contacts, family members, people who've been in the same meeting with people who are known to be infected, and treat them after they're exposed, but before they are infectious. That could be a very interesting way for early access for the antibodies in that type of patients, because what the rate of infection is, and you could get early clinical data. The doses that you're going to use for that kind of prophylactic study are unlikely to do any harm to anybody. And so the risk-benefit ratio there, I think, justifies some early use. In terms of, in the opposite end, might be patients who are intubated in an ICU, and it's unlikely. Generally, anti-infective antibodies are highly safe, and so it's unlikely there'll be any safety issues. But you might want to be a little more cautious in that population. So we think about those issues differently for the different populations. And like all early access programs, especially if the number of patients is limiting, as hopefully it will be when we get to the summer, then to the extent you allow early access, you might eliminate patients from participating in the trial. And so you want to do it in some kind of registry mode. So you can do it, I think, as you alluded to at the beginning, or as Scott alluded to, that you want to be able to make the drug available in some kind of registry so that you can provide it quickly to patients at the same time as you can collect data that will be supportive of moving the drug forward into larger populations.
Thanks, George. I want to thank all of you for a great discussion. Maybe turn just briefly to George Yancopoulos and Chad for just a final comment. We've only got like 1 minute, but any final thoughts you'd like to make sure you leave this group with?
Well, yeah, this is George Yancopoulos. I think there's nothing more important than doing well-controlled studies. I mean, whenever there are desperate people, unfortunately, they want to try anything, and the physicians want to try anything. That's why for our IL-6 receptor antibody work, we work really hard with the FDA and with everybody else involved to make sure that we get a real answer. We don't want to be giving false hope and putting out drugs out there that really don't work. We need answers. We need controlled studies. And I also just wanted to just communicate, because your question seemed to have missed the point that we're going to be in prophylaxis trials and in early treatment trials and in late treatment trials this June. With our antibodies. And that's just sort of a testament to the speed and power of our technologies. So we're talking about things that will be here and now that we'll be able to test in trials. And hopefully within a month or two of beginning those trials, we will have the sort of answers which will let the world know which of these approaches and therapies for which populations actually work.
Great. And Chad?
Yeah, I'll just echo that just time is of the essence here. I mean, this should be a Manhattan project, bringing together resource and collaborations that'll be required collectively to combat this virus. I mean, I've heard this term used, twice in the last week, once by my brother and once by Bob Nelson, to describe the situation that we're in. I really don't think it's an overstatement. We, meaning the collective public and private industry, can't afford to make this a linear process. I mean, we need to work with regulators, market access, and manufacturing to ensure that trials are expedited safely, medicines and tests are paid for, and that we can ensure proper supply upon success. And the FDA has been awesome and active in early and frequent consultation to identify and avoid any of the potential roadblocks to bringing both these effective diagnostics and therapeutics to patients. So and just wrapping up, I hope a lot of the work that we're doing as an industry coming together and throwing out some of the bureaucracies of normal business can continue beyond kind of this mode of crisis that we're in right now. So
great.
Yeah, let me just make one final comment on that. I think this has, in many respects, brought out the best of the industry. We have signed a number of collaborations with large companies, with small companies, where the business part of the discussions has been put aside and become secondary to how can we work together to facilitate bringing some of the patients quickly. If there's money to be made, which there may or may not be, we'll figure that out later. But let's make sure that we're prepared. And I've been in this industry a lot of years. I haven't seen those kind of interactions previously. So the industry really is coming together to do what we can.
Great. Thanks, George. And thanks all of you for your steps to bring the industry together and make progress for every stage in this disease, early, mild cases, moderate, severe cases, and as George Yancopoulos emphasized, prophylaxis as well. So thank you all. And actually that question about prophylaxis came from one of the contributions that we got from our audience. So if you do have a question for us that you'd like us to work into the discussion, it's hashtag COVID Therapeutics, all one word, COVID Therapeutics. But right now I'd like to go to our next panel on better and faster COVID-19 clinical trials. Just sticking with the Manhattan Project analogy, we've heard about steps at every stage of preclinical and clinical development to accelerate progress for COVID-19. Perhaps none of those is more efficient, more important than clinical testing, which can take significant time to set up the trials, enroll the patients, get the results, especially in a very dynamic environment like we're facing today and looking ahead in terms of active hotspots and availability of patients for participation in trials, given the potential candidates that could be coming into clinic in the summer and into the fall, we need to make sure we're using that clinical capacity, developing it and applying it as efficiently and effectively as possible. So this is a key part of the ACTIVE initiative that you heard about from Francis Collins earlier. And it's also a key part of activities from a number of other players in that effort and in other broad efforts to bring clinical trial capacity to bear and clinical evidence development to bear on the COVID-19 pandemic. So for this discussion, I'm very pleased to have with us Luciana Borio, who's currently Vice President, Technical Staff at InQtel. She's the former Director for Medical and Biodefense Preparedness Policy for the White House's National Security Council. It's been great to work with Lou over the years, and it's great that she's so involved in the COVID-19 effort. Also, Pamela Tennerts, who's the Executive Director of the Clinical Trials Transformation Initiative. That's based at Duke, but a collaborative that involves regulatory experts, FDA, and companies, product developers, other stakeholders from around the country and around the world on improving and transforming clinical trials. And also Robert Walker, who's the Chief Medical Officer and Director of the Division of Clinical Development at Biomedical Advances Research and Development Authority, BARDA, which is part of the efforts on preparedness and response in the Department of Health and Human Services, part of ACTIVE and very central more generally to our nation's efforts to respond to and prepare for public health emergencies. And if, Robert, if you don't mind, I'd like to start with you. Maybe you could give us the BARDA perspective on what we're seeing in trying to accelerate progress on addressing COVID-19, particularly with an emphasis on the effectiveness of clinical testing, but if there are other issues that have come up today, preparing for manufacturing capacity or other things that you'd like to comment on, please feel free to do so as well.
Well, thanks very much. Thanks for inviting me to participate in this panel. I'm really looking forward to it. Let me start by just saying that kind of how BARDA fits into the overall response structure. BARDA sits within the Office of the Assistant Secretary for Preparedness and Response. And our focus is on research and development of medical countermeasures that can be deployed in the event of a public health emergency, such as we're experiencing now. Our focus is really traditionally has been on late stage development, so phase 2 and beyond, what we call advanced development, and on securing products for the strategic national stockpile so that they can be deployed for the civilian population should that need arise. We, since our beginning in 2006, we have now supported 54 different products that have been either licensed or cleared by FDA, starting in 2007 with Sanofi's influenza H1N1 vaccine. And the primary role that we play in events such as COVID-19 is a across all of government coordinating function. And we do that primarily through the stand-up of the Medical Countermeasure Development Task Force. So the Medical Countermeasure Development Task Force takes an all-of-government approach and is saddled with the chief responsibility of coordinating across the USG and of ensuring that we identify gaps in development and come up with strategic solutions to how we're going to address that as an all-of-government entity. And the BARDA plays a lead role in that we chair and administer the task force. And we also run 4 of the working groups that are under that task force. So I'm a co-lead on the clinical trials working group with Matt Hepburn from Department of Defense. And then we also have a diagnostics, therapeutics, and vaccines working group. The membership on the task force includes BARDA, as I mentioned, but also other groups within the Office of the Assistant Secretary for Preparedness and Response, as well as CDC, Department of Defense, Homeland Security, FDA, NIAID, and USDA. And then we have liaison connections to other very critical members of the federal government response, including FEMA, Department of Energy, the White House National Security Council, and the Office of Science, Technology, and Policy. Centers for Medicare and Medicaid Services, and the National Guard. So you can see there's really a diverse array of just within the government stakeholders, all of which are part and parcel to this kind of coordinated response that we are charged with coordinating and leading. Under the clinical working group of the task force, the US government has now stood up 5 therapeutic clinical trials. So starting with the so-called ACT study, which is led out of the Division of Microbiology and Infectious Diseases under the US government principal investigator, Dr. John Weigel. That's a study that has enrolled over 1,000 individuals, was stood up from concept to first patient in approximately 3 weeks, which is probably unprecedented for the US government. And as Dr. Collins mentioned earlier, in a short period of time, maybe a few weeks now, we expect to have results from the comparison of remdesivir versus standard of care in patients who are hospitalized with COVID-19 and pneumonia. And that's going to be a critical data set for all of us that will inform how we treat and sort of how we move forward, both in the clinical treatment space and in the clinical research space. 2 other trials that are under this task force umbrella are hydroxychloroquine with or without azithromycin. The VA is in the lead on that one. And the hydroxychloroquine versus standard of care. Both of these are in hospitalized patients. And the lead on that is the National Heart, Lung, and Blood Institute. And then 2 studies that BARDA is sponsoring and that are being conducted by the commercial partners, commercial companies, both monoclonal antibodies directed against the interleukin-6 receptor. There are additional studies that are coming online, and we'll be hearing about them in the days and weeks ahead. But what I think probably the point to emphasize right now is the incredible degree of collaboration and coordination that's happening across the government. Particularly, I've mentioned already DMID and NIAID and what they were able to achieve. I'll also mention the FDA acting in, I think, probably unprecedented and extraordinary fashion to turn around applications, documents, protocols, clinical development plans in record time. We've seen documents come back within 24 hours. We've been meeting with them, and others like yourselves have been meeting with them as well on weekends and well into the night. They have recently stood up the Coronavirus Treatment Acceleration Program, which basically is a way to reach out to industry and others who wish to submit protocols and development plans for rapid comment by the FDA, and they guarantee a response within 24 hours. And so far, it looks like they've been able to achieve that. So I think it's an unprecedented time for all of us. What's been remarkable is the degree of collaboration, coordination across the United States government partners. And I guess we'll get into later in the discussion access to networks and sort of how to move rapidly as a collective organization in partnership with the private sector. But I think so far, we've been pleased with how we've been able to respond. So.
Great. Thanks very much for those comments, Robert. And Lou, I'd like to turn to you next. This is not your first, epidemic, pandemic, emergency response, but it certainly is an unprecedented one. And you have done a lot of work to try to facilitate the development of good clinical evidence when there's just enormous pressure to try to develop or provide access to treatments that might potentially help. Your thoughts about where we are and what's needed to make further progress in clinical testing and getting that evidence from here?
Sure. Thank you, Mark. And it's really great to be here. Thank you for inviting me. I think it's helpful to put things in perspective a little bit, how we used to do things before 2014, before the Ebola epidemic, where we would use a lot of these investigational products under a compassionate use mechanism. And then we would actually end the epidemic and having very little to show for, very little to know what really helped patients and whether we had hurt them in the process. In 2014, things changed with the Ebola epidemic, and I think the US government really took a very strong lead in pushing for what I call now a very radical approach. And the radical approach was that we should actually do RCTs in the middle of the emergency. And the thinking there was that they were just so powerful and really the fastest way to achieve 3 goals at once, which is the fielding of investigational products, if one considered that access can be given through a clinical study, especially when doses are limited, the detection of harm and detection of benefit. And what we learned too is that because it's such a powerful tool to detect those signals, it allowed everybody to move into the clinic much sooner. So we're willing to accept risks because you knew that if you randomize, you'll be able to detect those signals and minimize harm. And in essence, products went into the clinic sometimes after a dose was identified, both for vaccines and therapeutics. And I think that what we're doing today for the COVID-19 pandemic is very similar, but on on steroids, right? So there are many efforts underway that rely on pragmatic and adaptive RCTs that allow design in a way where success can be declared at the soonest possible time point. And those drugs then can be incorporated into the supportive care arm. I think it's important to keep into perspective that, in aggregate, we are suffering a horrific pandemic with a huge amount of lives lost, but that the mortality for patients, even those who end up in the ICU and intubated, and the intensivists are saying that they stay on for a protracted period of time, that the mortality is still in the single digits to no more than 20% in the US. We don't know for sure yet, but we're not talking about a situation where, 9 out of patients die and there's very little to lose. I think there's really, important to keep that in mind, that we have to be careful how we approach access to investigation and products, especially those for which, they can be game changers, but they can also cause a lot of harm. We don't know. I think the biggest challenge we face today is for using more RCTs. Of course, there's always the perennial one, patients are too sick to be enrolled. We can spend a whole webinar on that. But I think it's the fact that there's a perception that RCTs are slow. And I think that there's a real risk that they are slow if we don't do more to make them move faster. I think that we need to not lose sight that these trials have to be very pragmatic and simplified with broad inclusion criteria that really, are closest to how patients are presenting and using these therapeutics. I think that an area that is really very ripe for improvement or disruption, if you will, is data collection. Today's biggest electronic health record systems do not interface with clinical research software. And I think that manual data entry is still alive and well, and it's slow and prone to error. And I don't think that, you saw Dr. Collins mentioned and Bob Walker mentioned too, the NIH, the first initial phase of the NIH study that tested remdesivir over-enrolled, and the data is available. And we're thinking about, maybe a few more weeks before we know the results of the study. I mean, it's a randomized one-to-one study. We shouldn't have, wait for results for weeks if we could accelerate this component of data collection and analysis. And then lastly, I think it's important to also not lose sight that sometimes we think about how about the idea that we can incorporate tools that have been so important for other therapeutic areas, like surrogate endpoints or soft endpoints. And just because they're so valuable for those therapeutic areas does not mean that they are the fastest way for an emergent infectious disease. When you actually have a very hard endpoints available in a very readily available time frame. And so, the idea of a surrogate marker doesn't really accelerate development in any meaningful way. So we have to really keep pushing those messages. So I guess before to summarize, I think that we really need to redouble efforts to do RCTs. We need to find a way to bring clinical sites online to do pragmatic RCTs. That would be a Manhattan project. Improve the interface for data collection with EHRs. And like everybody else, I'm really hopeful that we'll have several blockbusters in the near future, because we desperately need them coming into the fall and winter. Thank you.
Lou, thanks for that overview. That's excellent. I know we're going to get into issues related to more rapid collection of standardized electronic data as we get into the next panel as well. It's a big issue there. But as you mentioned, Manhattan Project needed on recruiting more clinical trial sites that are able to work together because they're following the same platform, the same kind of protocols, and they've worked out ways to make their data and their methods comparable. This is what clinical trial networks are all about, and it's certainly part of the mission of the Clinical Trials Transformation Initiative. So Pam, I wanted to turn to you, and maybe you could talk a little bit about the efforts underway at Citi, Clinical Trials Transformation Initiative, both to understand what's going on in terms of trial protocols that are out there for COVID-19, and to try to drive more consistency, more of this network approach that we will have trial capacity when and where it's needed as a growing number of promising treatments reach clinical development. As Lou was saying, I mean, it doesn't seem like these trials should take that long for at least early stage to late stage patients. We're talking about meaningful outcomes that can be observed in days to a few weeks. And so it really does seem like a big issue is around getting enough trial capacity to do large enough, well-powered, well-designed, consistently designed studies, but that may need to involve multiple sites and a lot of collaboration around the country and around the globe.
Yes, thank you. And thank you too for having me on this webinar. I've learned a lot already, and I'm sure I'll be learning more as we hear the other speakers as well. So first of all, I think it's encouraging to see that clinical trials are actually getting off the ground so quickly these days. And that points to communication and collaboration and responsiveness of regulators and other people involved. But also, one of the groups that we haven't quite talked about as much is the actual sites executing the clinical trials. So if you think about it in these days, we've all seen the news reports about hairy doctors running around with masks, not enough masks, and trying to impose a clinical trial in there. You just want to be cognitive that you want to do something that makes sense for them as well, and that is streamlined. And the other thing I would like to say that was pointed out as well is we've seen a lot of things happening. IRB approvals in 3 days, regulators that respond in days versus weeks, amendments that are done in 24 hours. I sure hope that we can keep some of that momentum going afterwards, and we can sort of embed that in the other trials, because there's many diseases that could benefit from that. So what I wanted to talk about first is maybe a little bit of that efficiency aspect. And the Clinical Trials Transformation Initiative, I mean, we live and breathe improving clinical trials, both on the efficiency side and the quality side. So this is really what we focus on. And we have framework that could help with this called Quality by Design that I'll talk about in a little bit. But first of all, as far as efficiency, we have downloaded on April 2nd the whole data set of clinical trials involving COVID. At the time, there were 282 trials, 96 or so were interventional, so there were other observational and other kind of trials. And so, first of all, not everything is a good trial, potentially, right? So there's many different trials. There are very small trials. There's many different things. I'll talk a little bit about single drug trials. So at this point, nobody really knows what the winner might be. Hopefully there's multiple ones, but we don't know. So doing a single trial in public health emergency might make sense, but there might be an opportunity to look at master protocols. And master protocols is where you test multiple drugs at the same time within a network approach, and you can sort of do plug and play as drugs become available, or over time, things may look like options, or things may not work out and you can drop them from the trial. There are a couple of promising ones going on. The one thing we found on that same download is that there were about 25 different master protocols going on. That might be a little bit too much of a good thing, because hopefully you don't need quite that many. The WHO has one that they're starting, the Solidarity trial, but I want to just point out two of them that we were made aware of. One is a really elegant solution. It was already ongoing. It's called REMAP-CAP. They were looking at community-acquired pneumonia. And as this pandemic emerged, they were quickly able to add an arm that dealt with COVID. So this was an existing trial, the master protocol, and they just plugged in a COVID arm. This was an international effort, Europe, New Zealand, Australia, but also some US involvement. And the first US trial site is starting up at UPMC at Pittsburgh, where it's actually also including being master protocol, it's embedded in the electronic healthcare record. So it's really super elegant as a solution. You can do other things. In the UK, they have started the recovery trial. That trial went from first draft to first patient enrolled in 10 days. That is unbelievable. That is amazing. They used the quality by design approach to sort of look at things that matter. So they are also testing multiple drugs at the same time. And as of now, they started enrollment on March 23rd. They have 6,000 patients enrolled, so this can go quickly. It's a collaboration with the NHS over there. An aspect that is commendable and I hope will also lead to better trials is they are completely transparent. Protocol online, consent online, everything is online and everybody can look at it and just learn what they did and potentially draw some lessons from it. The other point I want to make is that by focusing on quality, you can actually create efficiencies. It will be improved. And all the rest I'm going to talk about assumes that we have a good scientific question, which in this case is, can we find a treatment? So that's hopefully the case here. That's not always the case in other cases. And then you can design a better quality trial that gets you that answer. And quality does not have to slow things down. As I said, this one master protocol was created in 10 days, so it doesn't have to slow things down. We need results, but you can only get results from with proper planning. And what matters is that sensible studies are done both with quality and efficiency. And when you think about clinical trial quality, that really depends on having a well-articulated plan that includes the protocol, the analysis, and the management. And it's really about focusing on things that matter and then taking care that you can support the quality in those critical areas. For example, on this webinar, I wanted to make sure my dogs were quiet, so I walked them before the webinar, that kind of a thing. And in protocol design, there's a couple of things that you can consider, and they're all listed in the principles document. For randomization, as Lou mentioned, is critical in this case. We really need to get answers, and a randomized, well-powered trial is the only way we can get there. And I won't elaborate on that because we've already heard a lot about that. Control arms matter too, because now their standard of care is probably a little bit different everywhere we go. Some hospital systems have included hydroxychloroquine in their standard of care arm at this point, whereas trials are trying to figure out whether that is a valid option, and then those trials could not happen in the systems where they've already included it in their standard of care arm. And we don't know yet what is going to work. Endpoints can be looked at as a forcing function for streamlined trials. We really need to look at endpoints that matter. This is a biphasic disease in some ways, where you have a milder set of symptoms for a couple of days, and then a subset of people go to pneumonia and ARDS kind of symptoms. So they might both need different endpoints at those times, and it just depends on which trial you're designing for which protocol that can then lead you to not measure things that you're not going to use. Because again, we're doing this in centers. They're not twiddling their thumbs waiting for us to come up with a trial for them at the moment. They're putting this on top of everything else we're doing. When we looked at our sample of 18 trials, and we looked at interventional recruiting, randomized trials that were not in China and Hong Kong, because on April 2nd, it felt like they were already on the way down, so that didn't matter quite as much. Overall, the average number of endpoints in our sample was secondary endpoints, so not primary. Luckily, those were like only 1 or 2. That makes good sense. But secondary endpoints were on average 13 per trial. Interestingly, for phase 2, it was lower, 7, than for phase 3 trials. Typically, that would be reversed, so that is just something interesting. And again, so we'll look further into that, but limiting the number of endpoints is probably not a bad place to sort of think about. Enrollment was also touched upon. A small trial of 30 patients is probably not going to get us a reliable scientific answer. It might give us sort of things to think about of maybe doing randomized clinical trials with. On the other side, if you are doing a trial that has an appropriate number of participants projected, depending on where you are and where the epidemic is at peaks or going down, you may or may not be able to do that in those sites, depending on where they're located geographically. And with that, I will turn it back to you. Thank you.
Great, Pam, thanks for that overview. Let me just add, for those of you who are interested in maybe doing more and participating in clinical trials, either from the clinical side or patients, Citi on its website, Clinical Trials Transformation Initiative, has some of these resources and tools available. And Pam, there's an upcoming webinar on Thursday that's going to get into best practices that you're doing in close collaboration with FDA and other regulatory experts. So something else to look forward to. And Robert, I know we're really tight on time. I just want to go back to you with one question, because I know BARDA is also working on the manufacturing issues that we talked about earlier. So trying to prep enough capacity for these advanced manufacturing products to make sure that that we're addressing that. Can you spend maybe a minute on some of the key things going on there?
Sure. So I think all of us are struggling with some of the same issues, which is we don't want to be in a situation where we have a product that looks like it's efficacious and we can't make it, or we don't have enough to treat the population. And what I can tell you at this point is that I don't have answers, but I know that BARDA and the rest of the USG team that we talked about earlier have assembled a rapid response group that has all the right types of people. So we've got clinicians and bench scientists and supply chain experts and manufacturing experts who are crafting a strategy now, which is how we're going to intervene at various time points along the development pathway and pull the trigger on various activities that will start the manufacturing campaign. There's calculated risks that have to be taken at every step along the way. We understand that. Not every product's going to be a winner. We have to make some guesses. We're going to be wrong a lot of the time. But I think everybody understands that we have to sort of march in that direction, and we have to start that activity now. So more to come, but just to know that BARDA, certainly the rest of the agencies with which we cooperate, are very much aware of the fact that this is a priority, and we need we need a better approach than we've had historically. And more to come.
Great, Robert. Thank you very much. Lou, Pam, it's been terrific collaborating with you on these issues related to COVID-19 and other topics. Thank you all for joining us for this event today. I'm sure there's going to be more to come. Right now, though, I'd like to switch to our third panel. So we've talked about all of the intense efforts that are underway to bring promising treatments through preclinical phases, and now these very challenging issues around clinical development and to some extent manufacturing, all these not well worked out, but certainly unprecedented efforts underway. Now we'd like to talk about the real world or post-market setting. As you've heard, with these intensive efforts, there will hopefully be good evidence, very good evidence brought to bear from these well-designed, efficiently done clinical trials and other learnings, background knowledge about products that are making it to market, some products that are already on the market, but almost certainly there are going to be important unanswered questions about these treatments. And looking at some of our hashtag COVID therapeutics, remember that's the place to send a comment or question that you'd like us to try to address. We've heard questions about effects in different subgroups of patients, vulnerable patients, those with complex conditions, the populations that have been disproportionately affected by the pandemic so far. We heard questions about potential side effects of some of these therapies that perhaps may not be fully understood in clinical trials, bringing them to market. And I think you've heard from everyone that there's an emphasis on developing maybe multi-treatment approaches, multi-pronged approaches, as has worked well in other viral infections here. All of those are issues that may not be fully addressed by the time treatments make it to market under these accelerated approaches that are intended to get strong evidence, but may leave some questions unanswered. So we want to talk about this real world side of evidence development, getting to more effective use of COVID-19 therapeutics in this last session. Very pleased to be joined by Amy Abernethy, Principal Deputy Commissioner at the Food and Drug Administration, like everybody at FDA, I'm sure going 24/7 these days on COVID-19. Jeff Allen, President and CEO of Friends of cancer research. And Jeff, I may have to change your title because you're doing a lot of work on COVID-19 research and development too, which we're going to get to hear about in just a minute. Emily O'Brien, one of my colleagues at Duke University, affiliated with the Margolis Center. She's also an assistant professor in population health sciences and helping to lead an important new trial through PCORNet, that acronym that you all heard about earlier, and I think many of you are familiar with, for large-scale, real-world clinical studies, including randomized trials in this case. And Steve Miller, the Chief Clinical Officer at Cigna, formerly Vice President and Chief Medical Officer for Express Scripts, who is leading some important efforts that health plans, that payers are supporting in the United States to try to develop better evidence. So I'd like to start with Amy. Amy, your job description before the pandemic included bringing much more effective, well-understood data to bear from a variety of settings that could potentially improve both clinical trials and post-market evidence. Here in this context, I know you've taken some important steps to try to adapt that strategy for COVID-19. So maybe you could just start off with some of the things going on at FDA in this space around real-world data, making it effective to use in real-world evidence, and we can go from there.
Terrific. Well, so first of all, thank you, Mark, and thanks to Duke-Margolis and Alexandria for having me today. You know, as you alluded to, I could comment for quite a while on how we might accelerate the data collection in clinical trials, as Lou was talking about before, or also how we could aggregate data sets as is being contemplated in the work with Francis Collins and NIH. But I'm going to focus on the approach that we're taking from the Office of the Commissioner at FDA right now, and really focusing on data sources that currently exist. So within the context of COVID-19, we've got this urgency to learn what we can as soon as we can. And that means that we need to be learning from the patients that are receiving care right now and trying to understand how do we apply that as quickly as possible. It certainly takes us out of our comfort zone at FDA, where we usually contemplate very careful work that has been carefully thought through and vetted, and we have also really thought through the precedent of what happens when we make decisions on real-world data and the decision itself, as well as the decisions for the future. But right now, in the context of COVID-19, we're really in a setting where we need to figure it out right now and acknowledge that this may take us out of our comfort zone. Practically speaking, with COVID-19, as we really learn from the care of patients, what we're seeing as electronic health record data is critical. And importantly, what we see is that because many of the patients are hospitalized, these are often data sets that are coming directly from electronic health records reflecting patients in the ICU, patients requiring oxygen, et cetera. The other thing that we're seeing is that it's really important that the data sets that we rely on and the variables that we're looking towards reflect also the critical data points that we're going to need to do analyses now and in the future. And one of the problems is that that's a story that keeps changing. So rather than sit around and wait until we learn how to use real-world evidence and make some of the hard decisions, one of the approaches that we've taken right now is to start learning from the data that are currently available. And what does that teach us about what decisions we can confidently make with the data that are available, as well as what we need to do in the future. So what do I mean by all of that? So first thing that we did was we said, what are the critical questions that we at FDA, as well as across the COVID-19 management community, have right now? Operational questions. Questions as it relates to epidemiology and clinical epidemiology, issues related to treatment and safety, issues related to, for example, how do we find patients to donate convalescent plasma or participate in clinical trials. Let's divide those questions into those that we can answer because data are currently available and we can go after the question in the next 2 weeks. What are questions that we can go after in the 3 and 12 week frame that require a bit of data cleanup, for example, new and analytic techniques, but the data are otherwise generally ready and we can answer those questions. And then also, what are questions that are going to take us 3 months more or so to answer? By dividing the questions in that way, what it's allowed us to do is figure out what data sources are already out there and how can we start to put them to bear. and also figure out how do we practice answering the questions. And it's taught us a number of things. So first of all, having a list of questions matters because it tells us what variables are needed, what kind of work needs to be done. Second, we've learned that we need to start agreeing on common data elements. Those common data elements should be in our real world data and also then should be transferred to what do we think are the common data elements needed for our clinical trials in the future so we can have some comparison between real world evidence and traditional clinical trial evidence. We've also learned that we need to develop common standards such as case identification and common covariates for our analyses. By doing this, what we've also figured out is how much we have to learn from each other. One group may be working on how do we clean up the electronic health record. Another group may be working on how do we do the right propensity score matching. And the only way to move this forward is to create the kind of partnerships that allow us to transfer information and learn together quickly, which takes me to partnerships. The other thing that we've learned here, and you heard this almost in every session, and it was really quite remarkable as we were listening to the therapeutic development section, is that most folks have put down whatever competitive barriers may have been in place and started to figure out how do we handshake and have partnerships that are starting to push this forward. So groups that have historically not been combining data sets, we've seen them combine data sets across lines. Groups that have historically not been thinking about how do we teach each other our own analytic methods have been teaching analytic methods. And those partnerships have been really key to moving things forward. Last thing I kind of want to describe are what are the questions we're answering right now, and give that as a highlight of what do I think is going to be possible in the future. We've used this critical question around hydroxychloroquine as what I call the platform for figuring it out. Practically speaking, hydroxychloroquine is being prescribed by many physicians in America outside of the context of a clinical trial. So that means that patients are receiving hydroxychloroquine or not, and their care is being picked up within the electronic health record. And if we ask the question, what do we understand about treatment patterns of hydroxychloroquine, about safety concerns with hydroxychloroquine, about potential treatment effect with hydroxychloroquine, trying to answer that question in multiple different data sets will teach us what data sets make that possible, how can we standardize the questions and the common data elements, what results do we find reliable and compelling enough, and also replicable enough against different data sets that we can actually answer potentially questions around hydroxychloroquine with confidence, but also then use that same platform approach for other questions. We're doing the same thing right now for drug shortages to try and understand demand and supply of drug, to think about how we're going to look at the real world performance of various diagnostics, including serologies, and also ultimately, can this help us develop essentially natural history cohorts that can serve as real world comparators for evaluation of, for example, drugs in the phase 2 setting in the future. So these are a few of the things that we're taking on right now with a sense of urgency at FDA. And I would highlight this role of partnerships, and I think you'll hear a little bit more from Jeff Allen around that.
Yeah, great. Amy, thanks for that overview of the FDA real-world evidence activities in this space. Sounds like they're growing rapidly. And one of those, Jeff, I will turn to you now about the work that you're doing in collaboration with the Reagan-Udall Foundation to accelerate the development of this kind of evidence. This is a new program, but one that seems to be getting some traction quickly.
Sure. Well, first, I want to add my thanks, Mark, to you and Lynn and the Duke and Alexandria team for putting together such a great and important program today. Yes, we have had the pleasure of working through with an effort that's being led by the Reagan-Udall Foundation and through an MOU process and in consultation with the FDA that we're proud to be collaborators with. It's actually launching today, called the COVID-19 Evidence Accelerator. And what we're hoping to do is really take several of the concepts that Amy began to describe and operationalize them as quickly as possible. And these are coming into two forms. The one is around what we're calling informal lab meetings, which most people on the phone are probably familiar with, at least at some point in time in their career, of having an opportunity to, on a regular basis, get together, informally share strategies, share preliminary results, and just tactically strategize around data presentations that are moving very quickly, and be able to just find a way to interact with experts and your peers to design the studies of the future. So these lab meetings are occurring once a week, and I think it will be a really great way to be able to bring experts together. We had our first one last Thursday and had 3 terrific presenters share some of their initial data, and we expect these to continue and think it will be a great data sharing environment to capitalize on the existing efforts that so many have described in this session and others before it. The second, in addition to the lab meeting, is around constructing accelerator parallel analyses. And really thinking about some of the things that Amy outlined in her discussion. We really started some of this work around key questions and data elements, and went out with the Reagan-Udall Foundation to be able to query really almost 40 different organizations and try and crowdsource a list of key questions to make sure that we were trying to bring in as much input and try and condense around a set of key questions, and then what the necessary core data elements would be in order to extract some information from electronic health records and other data sources to be able to begin to hopefully answer those questions. And one way that we're doing that is through the parallel analyses. We know that there are lots of great studies going on currently, and hopefully those will continue and answer very important questions that they've set out to do. But we're also hoping to identify secondary uses of some of this data. And while this isn't the same as conducting a randomized clinical trial, and it's not meant to be, it's really trying to leverage passively collected data on an already strained system, but information that can be extracted from health records and aggregated together through a series of different common analyses, common collection methods, and then common analyses methodology in order to bring results together faster. And the goal of this would be to interrogate an overall larger population, to provide rapid data on very different populations that may not be able to be done in the context of a standard clinical trial, and ideally cross-validate findings. So the first of these parallel analyses that we're looking to launch in a very rapid time frame is around some of the parameters that Amy just described around hydroxychloroquine use. And you can find more information on both the lab meetings, the key data elements and the key questions in the core data elements. These are available on a website. We put them up temporarily on ours at focr.org/COVID-19. You can access this information, learn a little bit more about the upcoming parallel analyses and the different parameters that we're exploring with a group of collaborators to begin that work. And then we've also put up a summary for that first lab meeting so that you can have some information around some of those early presentations. But I think the real goal here is just to try and leverage information that's coming out very rapidly. And hopefully through the accelerator, we'll be able to find secondary uses, create another hub for collaboration, have it be very data-driven and very rapid, and continue to work with a large group of stakeholders to be able to share this information in as quick real time as possible moving forward at such an important time.
Right, Jeff, thanks so much for that overview. Clearly some big opportunities for those kinds of secondary uses, this observational data analysis, but some challenges in getting there, which you're really trying to overcome around getting more consistency in the data standards and common analytic methods. So even if different groups are doing separate analyses, they're all kind of converging on the big questions in the right way. There's another effort that I'd like to talk about, and that's the CoreNet, which is a longer-standing effort to enable large-scale, both observational and now randomized studies in the real-world setting, and to introduce the CoreNet activities in the context of COVID-19, including a randomized study that is up and running now. I'd like to turn to Emily O'Brien. So, Emily, please go ahead.
Great. Hi, Mark. Thanks for the introduction and for the opportunity to be part of this panel today. So this is actually a really exciting time to be part of the CoreNet. I think the COVID-19 pandemic really presents an ideal use case to demonstrate the value of real-world data, and also to leverage prior efforts to develop infrastructure for evidence generation on a broad population level at this really unprecedented speed to meet the needs of patients and providers. There are a couple of key advantages that we're hoping to take advantage of and to use in our COVID work with CoreNet, and a lot of these have already been emphasized today and are sort of well known before COVID, but I think become even more apparent because of the need to generate high quality evidence quickly. So as Dr. Gottlieb and Amy both mentioned, we're dealing with scarce resources, and the use of existing data really provides the opportunity to reduce site burden and costs associated with data capture. And we know this from prior experience with trials that have been conducted in the network, like Adaptable, that have substantially reduced costs relative to conventional clinical trials. And the other thing to note is that many of the participating sites at this point have a lot of experience responding to queries for data, both for observational studies and randomized trials, through well-established mechanisms that have previously been pressure tested. The other advantage to networks is that they offer connections to highly engaged investigators who can provide leadership to help move things through quickly at the site level. And then, of course, the well-known sort of scientific advantages of use of real-world data, enrollment of less selected populations than are typically represented in conventional randomized controlled trials, that enhances generalizability, but also means that enrollment can happen more quickly. And as was mentioned earlier, because of the timing for initiating studies, there might be difficulty enrolling in the future if we see declines in infection rates, which really augments the need for access to a broad population of eligible patients. And PCORNet's composition is also, I think, really important to note that there are both academic and community hospitals that can really support generation of evidence that's relevant to patients who are seen in a variety of settings, and also support more rapid implementation of results in those same settings. Just a couple of key considerations that I wanted to emphasize that were also touched on by Amy, and by Pam earlier, that the need for rapid evidence, of course, can outweigh the need for high quality data. And we need the same attention to robust study design in the case of observational data, confounding control that we would require outside of the setting of a pandemic. And for real world evidence, this often comes down to a question of data availability. So is the required information available from participating sites to adequately address the questions of interest? And the appeal of large scale research networks is that many are already paying attention to data quality and have been for a number of years, and have developed really robust processes to evaluate foundational data quality, which can save time and effort for new studies that use those data. In PCORNet, for example, there's a lot of attention to data latency, which is critical to ensure that available information covers time periods of interest, especially in this rapidly changing environment. And one more thing to note that I think can really be of use here is that many of these networks also have established governance processes to efficiently move requests from investigators related to COVID-19 through review so that studies can get up and running quickly. So a couple of specific examples to PCORNet. Since it's a network of networks, it comprises investigators from 340 health systems and already has foundational data curation processes to support good understanding of the strengths and limitations of data to be used. For COVID specifically, PCORNet sites have created subset CDMs of patients that are based on COVID-19 status, as well as pseudo controls, for example, non-COVID flu that can be rapidly refreshed in order to support data characterization queries. And some of these data will be used to characterize a cohort of COVID-19 patients nationally and provide some information on demographics and pre-existing conditions. The network is also in the process of refining governance processes for fielding COVID-related research requests, which really support investigators who are interested in using the data to answer high priority questions. The registry that you mentioned, the HERO registry, is using this platform to understand the impact of COVID-19 on healthcare workers. So it's a large national registry that's open to any healthcare worker in the United States. And the goal is really to create a virtual community to connect healthcare workers with upcoming research studies, including those related to COVID-19, as well as to create a data set of risk factors and health outcomes for future analysis, so we can better understand the impact of the pandemic on this high-risk population. We're connecting patients based on where they tell us they're working with clinical trial sites, 40 clinical trial sites that are part of PCORNet already, and the goal is to enroll 15,000 healthcare workers in an upcoming clinical trial examining hydroxychloroquine as a potential prophylactic therapy for COVID-19 infection with follow-up at 60 days after baseline. We're really hopeful that we'll be able to generate evidence quickly for an important question in this high-risk group, and also have some information on patient-reported outcomes like anxiety, burnout, and depression that we'll be collecting as well to give us a more well-rounded perspective of the impact in this group. So in summary, I think this is a really great opportunity for us to leverage existing research networks to quickly answer questions related to COVID-19. Still certainly a number of important considerations to make best use of the available data and to produce the most robust evidence possible.
Emily, thanks very much for that overview. And it sounds like a facility with Picornet that can be used now for the hydroxychloroquine study, but potentially for other treatments as they become available, since you're building up this network of participants using digital tools and spanning across a very large number of organizations. That's great to hear about. I'd like to turn to Steve now with regard to the work that payers are doing in this space. So Steve, you've been involved in evidence development and assessing and trying to encourage effective use of new technology for a long time. It's a good background to bring to bear for the COVID-19 crisis, but maybe some comments you have about what, from a payer standpoint, seem like the most important issues for the new treatments that are going to be coming to market, hopefully with some good evidence behind them, but probably some questions where we need more evidence, as well as how you're looking at some of the treatments on the market now that are being explored for COVID-19 use as well.
Yeah, so Mark, thanks for having me, and Lynn and Joel, thank you for sponsoring this. This is really an important event. So as Mark said, we've been actually looking at some of these issues for quite a long time, and we got all the chief medical officers together from the payer community, or many of them together, initially actually to look at the supply chain, to make sure that we were being good stewards of the pharmaceutical supply chain, to make sure there'd be adequate supplies of drugs for patients as we move forward. The second big issue that then came forward was actually, how do we lower the barriers for patients to get adequate care? So what are we doing with rules that keep patients from getting to care? And so, as you know, we all suspended the copay when it came to testing so that patients could rapidly get tested. We've all suspended the copays for treatment so patients have no barrier to treatment. We then turned our attention to say, we have these data sets with our claims data. Can we look at our claims data and actually come up with some answers for things that people are asking about? Hydroxychloroquine, chloroquine, ACE inhibitors, ARBs, non-steroidals. The list of questions go on. And so we're now pooling our data to look at our claims data to see, can we see, while these aren't randomized clinical trials, but will these give us kernels of knowledge that actually can inform or even help us ask the right questions going forward? So we're encouraged with, as everyone has talked about, all the cooperation across the payer community, but also we're hoping that we can actually make a difference, adding some bit of knowledge to the other great things that Amy, Jeff, Emily already talked about.
Great. And Steve, thanks for that overview. Could you maybe say a little bit more about how you're actually doing this in the hydroxychloroquine and chloroquine context?
Yeah, so the first thing we've looked at was actually taking our populations with rheumatoid arthritis and lupus and saying, if they're already on hydroxychloroquine, is it working prophylactically to keep them from getting sick? And so hopefully we'll have a case-controlled trial that we'll be able to discuss actually this week. And so we're finishing up on that. But that's an example. You know, the trouble with it is it's not a true definitive trial, but it just gives you a kernel of knowledge to say, are these things actually working the way you hope they are, or are they not? And should we be looking in a different direction?
Mm-hmm. Great. Well, I want to thank all of you for these perspectives, and maybe Amy, I could come back to you. So we've got a lot of work going on observational data analysis, including what the Reagan-Udall Foundation is now pulling together in collaboration with FDA and a lot of outside participants. Got trial networks, real-world trial networks like Picornet as a leading example that are bringing resources to bear, and a clear payer interest in supporting steps to address important questions. You see more work that FDA can do, or FDA can do in collaboration with product developers, payers, these other stakeholders to help identify some of the important questions for real world settings and find effective ways to address them, either on the observational side or the clinical trial side?
So thank you. You know, as I reflect on all that I've been hearing, the first thing is that what we've been trying to do is organize the different data streams into a series of critical categories where ultimately the results then bubble up and get merged together in ways that we haven't historically thought about it. So, for example, there's the ways that we traditionally think about real-world data at FDA, such as the Sentinel Network. There are distributed analytic activities that can potentially teach us new information, such as the VA DoD system, the Odyssey network, what we're seeing, for example, with what we're just hearing about from Steve. There is the ability to generate information directly from data holders that we usually aren't used to talking to. And so we've got a number of activities like that going on at FDA. There's the work going on, for example, among pharmaceutical companies thinking about how to potentially pull together data sets in service of COVID-19. And then there's critical prospective work, such as what you've heard about from PCORNet. And what we've been thinking about is how do we bubble up the information coming from all of these different areas and start to put it together to look for consistent findings that provide enough signal that it should be that we should get it to the task force, to FDA, to CDC, to NIH in a consistent fashion. And also then what does that teach us about what's the next critical question to answer? Because I think part of the challenge here is recognizing that the questions are going to keep coming. And so we not only have to develop systems to answer those questions, but also systems to rapidly say, and here's the next thing we need to know. The example last week was as we started to understand the importance of acute renal failure, that taught us about what did we need to understand about drug shortages with dialysate. What do we need to understand about why we're seeing so much acute renal failure in the United States? And is it truly different than what happened in the rest of the world? What does this teach us about new therapeutics we may need to consider for the setting of treating patients who got COVID-19 and acute renal failure? And so that's an example of a question that's now bubbled up, and we now need to make sure that we're systematically thinking about how to address it.
Great. Thanks for that view of next steps. And we're just about out of time. I'd like to go to our other panelists for any quick final comments, thoughts that they'd like to add about, add to Amy's about how we make further progress from here and bringing together these different real world data sources, hopefully engaging patients and the public, healthcare providers and patients much more, as well as generating new evidence from randomized trials where needed. Maybe Jeff, to you first.
Well, hopefully, you know, our hope with the evidence accelerator is just to try and bring all of these different pieces together and create that very venue. You know, like you said, Mark, some of this is going to take some degree of standardization, and we're not trying to add to anyone's workload, knowing just how much is going on. But hopefully, this can be an ongoing process where some common elements across multiple different data sets can be leveraged to get to information faster. So it's a great opportunity. It's wonderful to see the collaboration that many others have noted at such a necessary time for this. So it's you know, it's a real privilege on our behalf at Friends to be working with the foundation, the Reagan-Udall Foundation, and with the FDA to be able to try and push this together, hopefully with many of you.
And Emily, hopefully more opportunities ahead, both for building on the PCORNet work and for answering questions that do need randomization.
Yeah, and one other point I wanted to mention is just that there's been a lot of encouraging progress on the informatics side with the WHO establishing the IC10 emergency code for COVID-19, as well as the new LOINC codes to indicate SARS-CoV-2 testing and the new RxQEs for remdesivir, for example. So hopefully we'll be able to establish that infrastructure that we need to answer questions now and in the future.
Great. And Steve, any last thoughts from the payer perspective, especially since so many of them seem to be coming together, just like we've heard throughout this entire webinar on different parts of industry coming together in an unprecedented way to address the epidemic?
Yeah, Mark, it's clear, and I've talked to you about this before. We're going to innovate our way out of this. And the innovation that's being done on the pharmaceutical and the biotech side is being mirrored on the clinical side. And so when you look at the innovations for real world evidence, clinical trials, and other things, it's extraordinary. So there's actually going to be a real silver lining to this pandemic, and that we're going to be left with capabilities we never had before and cooperation we never had before.
All right, well, still a lot more work to do, but an unprecedented foundation to build on. I want to thank all of our panelists on this panel, the previous ones, all of you for joining us today. This will not be our last word, just as we've got a lot of work ahead. We're going to continue our activities on advancing progress on therapeutics. Stay tuned to our website. If you're not on our mailing address at Duke-Margolis, please get on it. There will be more reports and more events like this one to come. But before closing, I'd like to turn to our collaborator and supporter in this effort, Joel Marcus and Lynn Zadowski from the Alexandria Summit. They've been very strong supporters in helping to bring together a lot of the experts from across industry to help inform our work. And Lynn, let me just end with turning to you for a last word.
Thank you so much, Mark, and thank you to the Duke-Margolis team. A very special thank you to both you, Mark, and Marianne Hamilton-Lopez for the leadership on this topic. We're honored to partner with you on today's important discussion. And thank you to my co-founder of the Alexandria Summit, Joel Marcus, who's the executive chairman and founder of Alexandria Real Estate Equities and the Alexandria Venture Investments. Big thank you to all of today's panelists and to everyone who's listening. We're honored to be part of the community on the front lines of the fight against COVID-19. Today, in partnership with the Duke-Margolis Center for Health Policy, we come together once again in the midst of one of the most profound threats to public health in a century. As of this morning, there were over 2.4 million confirmed cases worldwide and over 166,000 deaths, with nearly 762,000 confirmed cases in the US alone. That translates into around 30,000 new cases each day. This community of stakeholders virtually gathered here today has an urgent call to action, and it's inspiring to see the life science community mount an unprecedented response to the COVID-19 pandemic, rapidly increasing both resources as well as sharing information aimed at finding urgently needed solutions. As we referred to many times today, this is our Manhattan Project. So even though a vaccine is ultimately the most definitive way to arm the world's population against new waves of COVID-19 infection, therapeutics and prophylactic therapeutics represent an urgent and more immediate solution to mitigate the virus's impact now. So it's critical that we leverage our powerful collective voice to drive how we navigate this crisis and how we adapt so we can ultimately return to our new normal way of life. Together we'll get there, together we'll find a solution, we'll save lives. We look forward to continued discussions. As Mark said, it's not our last webinar, so stay healthy and stay safe. Thank you.
Lynn, thanks again to you and Alexandria. And again, my thanks to all of you who came together today. We've got a lot of work ahead, but it's great to see the foundation to build on and the unprecedented collaboration. We look forward to continuing to support that at Duke-Margolis. And just a last thanks to the whole Duke-Margolis team that helped bring this event together in collaboration with all of you. Hope you have a good rest of the day. Stay well and stay safe. Take care.